welcome to this explainer. Today, we're diving into what has to be one of the most awe-inspiring, action-packed sequences in all of human biology. We are looking at this truly epic, microscopic, 7-day hero's journey. We're going to trace the path from a single tiny cell all the way to the very beginnings of human life. Seriously, it's a story of survival against the odds, incredible timing, and some absolutely fascinating cellular machinery.
I mean, it really begs the ultimate biological question, right? How on earth does a single cell actually become the blueprint for a complex human being? Think about it. How do two microscopic entities even find each other in this vast environment, overcome what seem like completely impossible defensive barriers, and then entirely reorganize themselves into a perfectly functioning transport pod seeking a new home? It's this massive biological mystery, and we're about to unpack it step by step. So let's jump right in. So the whole journey kicks off with this absolutely crucial preparation phase. You see, when millions of sperm are deposited into the female reproductive tract, they aren't actually ready to fertilize an egg right away. No, they have to go through this process called capacitation. This involves some pretty extreme physiological changes. The sperm literally sheds specific proteins and carbohydrates, and the lipid composition of its cell membrane changes. Specifically, it dumps a lot of its cholesterol. On top of that, the membrane potential of the sperm drops from -30 millivolts to about -50 millivolts. All of this molecular shifting acts like a massive biological power-up. It makes the sperm hyperactive, which allows them to swim significantly faster toward that ultimate encounter.
Now, to pull this off, the sperm's anatomy is literally custom built for the trip. You've got the tail, or flagellum, acting as the motor. Then there's the midpiece, which is just packed tightly with mitochondria to generate all the ATP energy needed for this frantic swim. But the really interesting part is the head. Inside the head is the precious cargo, the DNA. But right at the very tip is a structure called the acrosome. Think of the acrosome as a microscopic lockpick. It's essentially a specialized payload of digestive enzymes. And as we're about to see, it holds the absolute key to breaching the egg's formidable defenses. But getting there is really only half the battle. Once those hyperactive, capacitated sperm arrive at the egg in the outer third of the fallopian tube, they hit a major wall. That brings us to part 1, breaching the egg's defenses. Picture the egg surrounded by this massive, thick outer fortress wall called the zona pellucida. First, the sperm binds to this shell. That physical connection triggers what we call the acrosomal reaction. The outer membrane of that lockpick acrosome we just talked about fuses with the sperm's own cell membrane, creating pores that release its payload. The sperm digests its way through the shell and actually penetrates the ovum. But right here is where the biology gets truly brilliant. The literal second that first sperm enters the cytoplasm of the oocyte, it triggers step 4, the zona reaction. The ovum instantly releases chemicals that harden that zona pellucida into an impenetrable fertilization membrane, literally pushing all the other sperm away. It's a permanent lockdown to prevent polyspermy, ensuring only one single set of genetic material makes it inside. And to get through that wall in the first place, the sperm uses a highly specific set of chemical drills. The acrosome releases hyaluronidase, acrosome, esterases, and neuraminidase. These specific enzymes basically work as a team to literally dissolve a tiny, precise pathway through that tough zona pellucida, allowing the spermatozoon to slip its genetic material safely inside the egg. Okay, so the fortress has been breached, shifting our story to this incredible moment of cellular fusion. The true moment of conception is happening right now in part 2, the first cell, zygote. Once inside the ovum, the sperm unpacks its 23 chromosomes, which then reorganize into what's called a male pronucleus. Now, the egg has its own female pronucleus, also waiting with 23 chromosomes. It's like a highly choreographed dance. Spindle threads called microtubules form between these two pronuclei and physically pull them toward each other. When they finally fuse together, they combine their genetic material to create a single diploid cell with a complete set of 46 chromosomes. This entirely brand new, unique genetic blueprint is the zygote. It is literally the very first cell of a new human life. But obviously, a single cell can't build a complex organism all on its own. Now that the blueprint is finalized, the pace of our microscopic story shifts into overdrive for part 3, the Great Divide. Let's actually anchor ourselves in a timeline so we can see how fast this week-long quest moves. Day 0 marks the formation of our zygote. Almost immediately, it kicks off a process of early cell division called cleavage, which is driven by a mitosis-promoting factor. By day 3, it's already multiplied into a 16-cell cluster known as a morula. Fast forward to day 5, and it transforms into a much more complex structure called a blastocyst. And by days 6 and 7, this tiny little entity is ready to begin the final, most crucial step of its initial journey, implantation. Let's actually zoom in on that day 3 morula stage for a second. The cells in there, they're called blastomeres. They aren't just haphazardly dividing into some loose clump. To survive, they have to perfectly organize. They undergo a critical process called compaction. This is where those blastomeres become super tightly adherent to each other, forming tight junctions. It basically solidifies the entire structure of the developing embryo, preparing it for all the specialization that's about to happen next. And remember, this whole time, the zygote isn't just sitting still, waiting around. From the exact moment of fertilization out in the outer ampulla of the fallopian tube, these dividing cells are constantly on the move. There are these sweeping motions from tiny hair-like cilia, and that's combined with the muscular peristalsis of the fallopian tube itself. Together, they are constantly propelling the developing morula and early blastocyst along the tube, moving it inevitably toward the uterine cavity. By the time it arrives in the uterus around day 5, it's a full-fledged blastocyst, and its cells have basically separated into 2 totally different teams with distinct destinies. On the outside, we have an outer ring of cells called the trophoblast. Think of them as the support team. They're going to go on to form the future placenta and the chorion, and they immediately start secreting the hormone HCG. But tucked away safely on the inside is a cluster called the embryoblast, or the inner cell mass. This precious inner cluster is the main event. It's what will actually form the entire embryo itself, along with the amnion and the umbilical cord. Okay, so our blastocyst has made it to the uterus, packed with a specialized outer layer and an inner embryo blueprint. But there's a problem. It's still trapped inside that original protective shell, the zona pellucida. It's time for a major transformation in part 4, hatching of the blastocyst. Now, you might actually be wondering, why did the blastocyst even keep that shell this whole time? Well, it turns out it acted as a completely vital protective transport pod. First, it kept those dividing blastomers tightly packed together, so that compaction we talked about could happen. Second, it acted as a crucial immunological barrier against the mother's immune system. But most importantly, it physically prevented the embryo from implanting prematurely while it was still traveling through that narrow fallopian tube. If it had implanted there, it would result in an ectopic pregnancy, which is highly dangerous. But now that it has safely reached the spacious uterus, that shell is totally in the way. The blastocyst literally has to hatch. Those outer trophoblast cells start producing enzymes that finally digest the zona pellucida from the inside out. The blastocyst essentially sheds its protective shell, emerging totally vulnerable, but finally ready to attach to its brand new home. With the shell gone, the hatched blastocyst has only a very short window to secure a lifeline to the mother, or it just won't survive. This brings us to the final, desperate step of our journey, part 5, implantation in the womb. So right around day 7, the newly hatched blastocyst rolls along the endometrial lining of the uterus and finally adheres to the epithelium. And check this out, it specifically positions itself so that the inner cell mass, the embryoblast, is facing closest to the endometrial wall. It is literally burying itself into the thick, nutrient-rich lining of the uterus. And honestly, the way it digs in is surprisingly aggressive and highly orchestrated. First, you have the adhesion. Then, those outer trophoblast cells go entirely on the offensive. They secrete hydrolytic enzymes that break down the uterine tissue, allowing the blastocyst to actively burrow deep into the functional layer of the endometrium. As they dig down, they actually erode maternal blood vessels, creating little pools of blood called chorionic villi. This establishes the very first fetal-maternal blood exchange that secures the absolute nutrient lifeline the embryo needs to survive and eventually completes its total embedding right into the uterine wall. Now, the mother's body is completely on board with this miracle, adapting gracefully to sustain it. The endometrium physically transforms into what is now called the decidua. The decidua basalis forms right at the base of the implantation to become the maternal side of the placenta, while the decidua capsularis actually grows completely over the embryo, enveloping it safely within the uterine wall. The mother's body has totally adapted to welcome and protect this tiny cluster of cells. When you actually step back and look at this entire sequence, from capacitation and those tiny chemical drills of the acrosome, to the beautiful genetic fusion, to the aggressive, perfect timing of implantation, it is just staggering. Against all odds, this incredibly perilous, microscopic 7-day journey perfectly aligned to create the foundation for a human life. It really leaves you with a profound sense of awe, and honestly, it begs the question, what other microscopic miracles are happening inside of us right now, entirely out of sight? Thanks for watching.